In glycolysis, the glucose in the cytoplasm is broken into two 3-carbon sugars known as pyruvate by 2-ATP, which phosphorylate it and release ADP. NAD+ is then reduced to NADH by collecting 2 electrons. The molecule then undergoes substrate-level phosphorylation, in which the phosphate group is broken off to combine with ADP, producing 2-ATP.
After the pyruvate is made, it moves through the outer mitochondrial membrane into the inner mitochondrial membrane. Then a carbon atom splits off to form CO2, after which NAD+ takes 2 electrons from the newly made acetyl, becoming NADH. The acetyl combines with coenzyme A to become acetyl-CoA.
In the cristae, acetyl binds to oxaloacetate to produce citrate. Citrate is later converted into isocitrate through the removal and addition of H2O. The oxidation of intermediate molecules produces 2CO2 and NAD+, which is converted into 3-NADH as products. 1 ATP is gained through the combination of a phosphate group and ADP. 2 hydrogens from the intermediate molecule are transferred to FAD to produce FADH2 through an oxidation reaction. This cycle occurs twice to process both pyruvates, thus resulting in twice the amount of each product.
The first step of oxidative phosphorylation is the electron transport chain located in the cristae. The NADH produced by glycolysis is oxidized into NAD+, releasing 2 electrons to protein complex 1, where it passes through a flavoprotein and iron-sulfur protein. The electrons pass from ubiquinone or CoQ to the rest of the protein complexes, or cytochromes. Meanwhile, hydrogen ions are pumped against the concentration gradient and into the intramembrane space. Complex 4 then passes the 2 electrons to an oxygen, which bonds to 2 hydrogens, releasing H2O. FADH2, produced in the citric acid cycle, adds 2 electrons to protein complex 2, where they pass through to complex 4 and bond to an oxygen. As a result, the electron transport chain provides 1/3 less energy for ATP synthesis when the donor is FADH. In chemiosmosis, a protein complex called ATP synthase makes ATP from ADP and PI. The electron chain uses exergonic flow to pump H+ ions across the membrane into the mitochondrial matrix. These protons bind onto the ATP synthase rotor one by one, causing it to combine ADP and PI. This H+ gradient is referred to as a proton motive force. Overall, cellular respiration produces about 36 to 38 ATP total.
In alcoholic fermentation, glucose is converted to 2-pyruvate, which releases 2 ATP. The pyruvate is then converted to acetaldehyde by releasing 2 carbon dioxides. Then acetaldehyde is reduced by NADH to ethanol. This then regenerates the NAD+, needed for the continuation of glycolysis.